Engamana, engareo, engaro rangatira, onge hoafa, tena koto, tena koto, tena koto katoa. My name is Bryony James. I'm the provost here at Te Herangawaka, Victoria University of Wellington, and it is my very great pleasure to offer a warm welcome to all of you here this evening, a warm welcome to family and friends, to distinguished guests, to colleagues, both staff and students, and to our wider community. Welcome to the inaugural lecture of Wayne Patrick, Professor of Biochemistry in the Faculty of Science. Inaugural lectures are a fascinating little quirk of the traditions of universities. When we ask somebody who's probably given hundreds, if not thousands of lectures during their career to give their inaugural lecture. Because this is us welcoming our newest professor to our ranks and inviting them to give their first lecture to the public as a professor, having achieved what is actually the highest level of academic rank that you can. So congratulations are always in order, but also it's a welcome time for us to come together and celebrate the breadth of experience that we see in our academics. So it's my very great pleasure this evening to introduce our newest professor. Now, the tradition of these introductions is there should be some little familial anecdotes, some jokes, personal touches, but Wayne's been fairly unforthcoming in providing such material. And I was going to ask his boss, Professor Nikki Nelson, but she said she's got plenty of dirt. She's saving that for the vote of thanks. So you've got that to look forward to at the end. So I will limit myself more to a little review of Wayne's career to date. Professor Patrick gained his BSc from the University of Otago, and then won a Prince of Wales scholarship to do his PhD at Cambridge. Following that, he went to the US and spent 4 formative years as a postdoctoral fellow at Emory University in Atlanta, where he learned to love American football and the Atlanta Falcons. I must say, not the winningest team of the NFL. After 8 years overseas, Wayne returned to New Zealand. He bounced around some of the lesser New Zealand universities before he joined us here at Vic in 2018. And here he leads an interdisciplinary research group that addresses fundamental questions about enzyme evolution and why life is the way it is. So just the small questions. The group also used their fundamental insights to engineer enzymes for applications such as biosensors and better RNA vaccines. So actually, we've all got something to be thankful for there. Wayne modestly attributes his success to the 60-plus students and staff from 15 different countries who have trained in his group. This includes 13 PhD students, 8 master's students, 16 honours students, and no fewer than 9 postdoctoral fellows. He's supported the work of this group and himself by bringing in what is frankly an eye-watering array of external research income, including a Rutherford Discovery Fellowship, 4 Marsden grants, and 3 MB Smart Ideas grants. Now, as an ex-Deputy Vice-Chancellor research and naturally nosy person, I see that array of externally funded research and I think, well, what's he done with it? Let's have a quick look at his publications. So you do the academic equivalent of Google stalking and you jump on Scopus. And I must say, I was relatively surprised to find only 13 journal articles that had only been cited 128 times. It turns out Patrick Wayne is a grad student in Albuquerque, whereas Wayne Patrick has a rather more impressive array of 64 articles that have been cited well over 3,000 times. And whilst doing all of that, his intellectual curiosity and creativity means that he's also been the lead inventor on 5 patent families, and alongside a former PhD student, he's currently working with Wellington Uni Ventures to spin out a company based on biosensor technology. Now, obviously, it's not just us here at Vic who have recognised his contributions at the interface of fundamental biochemistry and applied biotechnology, as he's also been recognised with several prestigious awards, including the Research Excellence Award from the New Zealand Society for Biochemistry and Molecular Biology, and he's previously been named New Zealand's Young Biotechnologist of the Year. Now, Wayne is not just an impassioned researcher. He has taught extensively at all levels, and I commented as I was on the way here, obviously a lot of students here this evening, and that is an incredible accolade for an inaugural lecture. So thank you to the students for coming. Universities are places where knowledge is created and shared, and Wayne shares his love for the power of biochemistry to answer the big, important biological questions by finding creative ways to connect his students to molecules that they can't see. He aims to empower all of our students with transferable skills for their future careers. An example being his molecular modelling assignment, where he employs cutting-edge 3D modelling software and virtual reality so that students can appreciate and experience protein structure and biomolecular interactions. As you're likely realising, if you didn't already know, Wayne wants to push forward all aspects of his discipline. And I think a great example of this was when he was president of the New Zealand Society for Biochemistry and Molecular Biology, when he wrote and introduced the Society's equity policy, making it among the first of New Zealand's life science societies to formally commit to diversity and inclusion across all operations. Similarly, here at the university, he doesn't hesitate in taking on leadership roles. He spent 3 years as the director of the Centre for Biodiscovery. He's been deputy head of the School of Biological Sciences twice, I believe. And now he's stepped into the role of Associate Dean Research for the Faculty of Science. And he is as passionate a champion for the research of others in his discipline as he has always been for his own discipline throughout his career. If, like me, you're a nosy Google stalker and you jump onto his website for his research group, you'll find pictures of all of them out there tramping and playing mini golf. So it's a pretty clear thing that Wayne's enjoyment of the outdoors and trail running and tramping is having as much influence on his friends and colleagues as his research is. And it is this enjoyment and love for his work that we are looking forward to hearing more about this evening, as Wayne takes us through his inaugural lecture, Enzymes I Have Known and Loved. He will try to explain his decades-long obsession with enzymes, and he will introduce the insights from his research group that come at the interface of biochemistry and evolution. And he will argue that enzymes should become New Zealand's ultimate high-value export. It's my great pleasure to introduce Professor Wayne Patrick.
Thanks, Bryony. I'm kind of reminded of that Toby Keith song, Ain't as good as I once was. Nevertheless, let's get started. It's quite a day to give this inaugural lecture. I woke up this morning, of course, thinking about having to give a public lecture to a diverse audience about protein structure, protein function, protein evolution, protein biotechnology. Then I logged on to the news websites and found that the Nobel Prize for Chemistry has just been awarded in those disciplines today. So I hastily changed a couple of slides. I will say, though, that whilst the ending now involves this new flavour, my sleepless night last night wasn't because I got the midnight call from Sweden, so spoiler alert. Instead, I thought I'd start this lecture here with my official PhD graduation photo. As good a place as any, I think, to start the story. If you've watched really carefully, I've already stripped off some layers, partly because I thought it would be stuffy in here, but mostly because that's like the nice ceremonial eye candy. But actually, this is the gown that I wore on this day in this photo, and so struck me that it's kind of a bit battered now after lots of formal halls at lots of Cambridge colleges. It's kind of old and tired, and so it seemed appropriate that I should roll it out for an occasion like this at this point in my career. The other thing that I had in my suitcase, the other artifact that left Cambridge with me, was this PhD thesis there on the right. Large, some might say self-indulgent, though I like to point out that it was only single-sided printing in those days. And it contained a huge amount of data and information about two enzymes, alanine racemase and phosphoribosylanthanolate isomerase. Emily's here, she already knows where this story's going. Parker. There's two Emilys in that line of sight. Putting this lecture together made me realize that really was the start of a long and complicated love affair with these protein molecules called enzymes. And so I started by looking at my own CV, Google stalking myself, Bryony, and trying to compile the list of all the enzymes that me and my group have worked on since my PhD. I know this is kind of already the part where the biochemists in the audience are looking at this list and thinking, oh yeah, that one, yeah, that's a good one. One of my favorites. Of course you did. Of course you did. But everyone else, who I appreciate as most of you, are like, come on now, come on. It's slide 3 and you've lost me already. And not only that, but these are clearly not real words. And so we all know there's no such thing as a sulfhydrolase. But my goal for the next 40 minutes or so is to try and convince you that in spite of their ridiculous names, these enzyme molecules are fascinating, they are worthy of study, they inspire quite fundamental, thought-provoking questions about the nature of all life on Earth, because as we're going to talk about, every cell and every organism is packed full of them. And not only that, but in the last part, talk about this idea that knowing more about enzymes really presents this interesting and unique economic opportunity for New Zealand. So dovetailing into some of the biotech stuff that we've been doing over my time here and previously. So I do think no matter whether you're a biochemist or not, you've probably run into enzymes in your day-to-day life. So for example, you may have something like this in your laundry. Obviously, this is Persil's best powder because it's the one that's got enzymes in it. As it says on the ingredient list, they have some role in breaking down and removing stains. So already hinting at some of the magical powers, the mind-blowing functions that these molecules can have. They're way more interesting and cooler than just being able to break down and remove the stains in your dirty clothes, though. As I've alluded to, every cell in every organism, including every cell in your body right now, is busy doing hundreds and hundreds of chemical reactions. Molecules are being made and broken constantly in front of my very eyes. And the only reason those reactions are happening on a time scale that's remotely compatible with biology, with life as we know it, is because they're being accelerated by enzymes. So enzymes are really the proteins inside cells that catalyze reactions, take some really tricky chemistry and bring it into a timescale appropriate for life. To give you an example, one of the ones I teach about in Bio 111, there's an essential chemical reaction for making the building blocks of RNA, one of the key molecules in our cells. With no enzyme, if we just waited for that reaction to happen, it would happen about once every 74 million years or so. And so life's not going to start that way, right? There needs to be something that accelerates those reactions in order for all of the functions, the molecular functions that constitute life as we know it. That's why the functions of enzymes have fascinated me for my entire career. Those functions are determined by their beautiful and intricate structures. And so hopefully if Tiff and Matilda and Jordan are here, they'll recognize these figures and be intimately familiar with these particular structures. They're made, these enzyme molecules or proteins, they're made as chains of amino acids. There's a 20-letter alphabet of the different types of amino acid, and that determines how those chains fold into these, as I say, these beautiful and intricate structures, whether they curl into those helical structures, form sheets or loops or strands, in three-dimensional space, like the picture on the left there. You see on all the lumps and bumps, the negatively charged parts, positively charged parts, the parts that like water, the parts that want to be away from water. And that determines their function. And really, at the heart of an enzyme structure, and therefore its function, is that intricate fold places just a small number of the amino acids in exactly the right place that enzyme will recognize one or two substrate molecules, one or two chemicals inside the cell, and put them in just the right environment for a reaction to happen at a speed that's consistent with biological processes. All of this, of course, is happening on a scale that's much too small to see with the naked eye, much smaller than the white powder in here. the nanometer scale. And so, as you can tell, by the time I'd got to the end of my PhD, I'd really drunk the Kool-Aid about how, the awesome power of enzymes. I had a look at these 3 biochemistry textbooks that were all in my office last week, and they've all got statements along similar lines about the characteristic features that define enzymes. The one here I've pasted at the bottom is from the only one of these that has an ebook, Garrett and Grisham, that says characteristic features of enzymes are catalytic power, specificity, and regulation. They can do one job really, really well. They catalyze one reaction with speed and precision, and they do it exactly when the cell needs it. When the cell doesn't need it anymore, that enzyme gets turned off. The whole rest of the network functions until it's needed again. I was all into that by the end of my PhD. But there was a question that puzzled me, and that was the evolutionary one. So if all of these enzymes are such powerful and specific catalysts that do one thing and one thing only, where do new ones come from? Are new enzymes born that way? Are they born sort of as these beautiful kind of the pinnacle of protein evolution? Seems unlikely. We knew, even back in my PhD days, that new enzymes could emerge in biology really quickly because, we've spent the last century or more introducing human-made chemicals into the environment. So we knew already, even in the 90s, that if you introduce a human-made pesticide or insecticide into the environment, maybe in a decade or so, the soil bacteria will have figured out how to eat that for breakfast. They would have evolved enzymes that could metabolize that chemical. Similarly, and perhaps even more frighteningly, I suppose, the phenomenon of antimicrobial resistance. Here we've got a whole bunch of different antibiotics on these little white filter paper discs on a lawn of bacteria. You can see that in some cases the antibiotic works and the bacteria can't grow up to the edge of the disc with the antibiotic. But in the ones down the bottom, the bacteria don't care at all that there's an antibiotic in that filter paper disc. And the reason they don't care about it at all is that they've evolved enzymes to break down those antibiotics. So enzyme evolution can be rapid and have these, transformative and important effects. But at the end of my PhD, there was really unanswered questions about what was the evolutionary process that gave rise to this and how could it happen so quickly in biological systems. I was really fortunate to spend this time at Emory University, like Brian mentioned, working with a very key mentor in my career called Ichiro Matsumura. And so he was also puzzled and fascinated by this problem. And here's one of the first in his own postdoctoral time to discover and characterize a so-called promiscuous enzyme. So he found an enzyme that had evolved to do its job in a cell, but he figured out that if it was given a different substrate, a different sugar instead of the one it normally nibbles, just a little bit low activity on the side, it could break down that other sugar as well. So it had this kind of moonlighting, it would slip out and have this secondary function. And that made sense in the context of kind of standard evolutionary models about how life might have begun. The very first enzymes on the planet in the primordial soup. There'd been the sense that they probably, when life first arose, they were probably multifunctional, sloppy, did a bunch of different reactions, and through regular old kind of evolutionary processes of gene duplication and divergence, you'd evolve families of more specialist ones. So ancient primordial enzymes, sure, we had a model that said they were likely to be slow and sloppy. But Ichiro had found a modern enzyme that had vestiges of that same kind of those properties. And so the outstanding question in the field then was, how special was Ichiro's enzyme? Was that just a one-off fluke that we found a modern enzyme with this ancient primordial kind of property to it? I should also say that Ichiro was relatively eccentric, as you could tell from his official staff photo on the departmental website in those days. I should point out that his vision is perfect. The glasses are completely for comic effect. But it's also full of these pearls of Zen-like wisdom. And so I arrived in his lab. I was like, yeah, love your work. You know, I've spent the last 3.5 years in Cambridge studying this enzyme called phosphorybosyl and threonate isomerase. I've got this great idea to look for enzymes that have promiscuous PRAI activity. And Chiro said, Wayne, I've found one enzyme. You need to find all the enzymes. I was like, but Chiro, it's impossible. The techniques for doing that won't be invented until 2024. But sure enough, we thought about experiments that would try and generalize this finding. And so, you know, as a mentor, he was great at getting me to think big and think broad and think fundamental. And I was also I was like, but the other problem with Chiro is that even if I do manage to do this, it'll be this real kind of skim over the surface. You know, I'll find lots of examples if it all goes well, but I won't be able to explain them all in the beautiful biochemical detail that I know and love. It's like, don't worry, Wayne. Take the low-hanging fruit. Others will fill in the rest of the details. And he turned out to be right. And so eventually, after thousands of hours in the lab, I figured out how to do the series of experiments to really looked at how common and broad this property of promiscuity was in all the enzymes from the lab bacterium E. coli. So I did kind of over 100 experiments. And the big reveal was that it was really, really common. And that was surprising at the time. Now it's that's the thing. But it really kind of led to this new view of enzymes that in over 20% of my experiments, I could remove one enzyme from E. coli and find a promiscuous one from somewhere else in E. coli that could take its place because I had this little bit of side activity that would become essential in the absence of the one I'd removed. So it really led to this growing sense, this new view of enzymes that wasn't like the one in the textbooks. You know, the textbook view was and is this idea of beautiful specificity and high activity. But instead, my work in Chiro's lab helped to craft this new narrative that there was messiness and noisiness and sloppiness everywhere, that the metabolic networks that we'd memorized as undergraduates were way too simple and way too clean and tidy. The other formative thing that happened to me in my Emory years was that I asked Monica Girth to marry me. and so about 2 minutes before this photo was taken, when we propped our first digital camera on that rock in the foreground. And so I do want to point out that I've really been privileged and honoured to live the dream of every nerdy science boy. You know, not only did I get to marry the cutest girl in the lab, but we designed experiments together. We wrote papers together. As our career's gone on, we've won grants together. For the last 10 or 12 years now, we've run a joint lab group together. It really has been, as I say, an amazing privilege. It's only just been a few little kind of arguments over the kitchen table around the best protocols to make electrocompetent cells and that kind of thing. But overall, if this isn't the secret to a happy marriage, I don't know what is. So I came back from the US full of these ideas, controversial ideas, textbook-breaking ideas. The textbook's all wrong. They don't know anything about enzymes. And so set about establishing my own independent research career. And I really do, at this point, want to stop and say that nothing I'm going to say for the rest of this talk is really done by me. So from here on in, it's been the other great privilege of my career to have had this team of amazing, enthusiastic, smart, energetic scientists work with me. Students, postdocs, research assistants, international visitors. So if you come and visit me in my office, the history of Team Enzymes is there for you to see, whether it's in the list of the compiled set of theses on one shelf with a small amount of creative license taken with Jordan Compton's here. It's at the printers right now so that she can walk across the stage and graduate in December. I'm not sure if pink was a colour choice, but if it was, she would have taken it. It's been a long-standing thing in my group that every fortnight we have meetings in which everyone comes along with a little printed out set of bullet points about what they've been up to, and my real-time history of the lab is therefore contained in these ring binder after ring binder of every lab report that's happened since I first got a group. And of course, there's the top shelf where all of the major events in the history of my lab group have been commemorated. If you're worried about health and safety, Bryony, which I'm sure you are, I'll just point out that the photo up there was taken on Sunday after the earthquake on Sunday morning, and so, in fact, the earthquake restraints on the front of the shelf work fine. Here they all are, in order of appearance in my group. Those of you who are in my group, who are in the audience, will be frantically looking. Don't tell me if I've forgotten someone. There's 66 names on that list, and so like any true academic, I'm going to take the next 66 hours to do justice to their work. At least I should, but of course I don't have that much time, so instead I think I have to apologise because I'll do justice to none of their work in the way that it could or should be done. As I say, though, the main point is that it's been this huge privilege to have all of these people work with me over the years. I'll give a few little snippets of the kind of stuff that we've done. And I'll start with my first PhD student, Valerie Sue, who really took what I'd done in a Chiro's lab to the next level. So she had this blockbuster PhD. Good advice for early career researchers: make your first PhD student a really good one. Sets a high bar for those who follow. So she looked for promiscuous enzymes, but that could do something novel. So I'd said, I'm going to remove an enzyme from the metabolic network and try and find one to take its place. She said, I'm going to look for promiscuous enzymes that give a bacterium a little head start on the path to antimicrobial resistance. And so effectively, she took a non-pathogenic lab strain of E. coli bacterium and exposed it to every antibiotic we could find, about 240 different antibiotics, and asked it to evolve resistance by using a promiscuous enzyme that didn't evolve to do that job because this lab strain had never seen these drugs before. And the results were kind of horrifying. So in almost 40% of cases, a non-pathogen like E. coli already had kind of the functions inside it to give these first little steps towards full-blown resistance. It wasn't full-blown resistance like I showed you before. It was a matter of kind of growing at slightly higher concentrations of a drug than it could without those promiscuous enzymes. But nevertheless, if you weren't nervous about antimicrobial resistance before, you should be now, because it's really easy to arise in bacteria based on just the enzymes they've got inside them already. At the same time, students starting with Matilda started doing that deep dive into the real kind of mechanistic and structural aspects of promiscuous enzymes that I'd longed to do in a Chiro's lab. And so Matilda's PhD was really unprecedented at the time in its depth. So she had a case in which collaborators had evolved a different bacterium, removed one enzyme and evolved it so it had to use another enzyme to take its place. And there was a series of changes in that enzyme, mutational steps involved in that process. So Matilda did this amazing deep dive into exactly how every one of those changes in the protein decreased the old activity, increased the new activity, how those traded off. Not only that, we solved structures of all of the enzymes, so at absolute atomic level resolution, we could explain how a change in the evolution of a bacterium had manifested at a molecular level. And it was all in the context of the evolving bacterium, how these mutational changes caused it to grow faster or slower in a bunch of different environments. So this real kind of blockbuster in her case at connecting all the dots between evolution all the way down to kind of atomic level explanation of how these molecules work to affect that evolutionary outcome. We did a bunch of those kind of studies through those years with the other people listed on that slide as well. But one thing that I kept noticing and became really obvious is we're doing all these evolution experiments, changing the functions of enzymes, making bacteria evolve new ones. But they'd never evolved them to be as good as the enzymes in the textbooks. And so that evolved to a point where, of course, the enzyme was good enough that the bacterium grew fine. The bacterium didn't need the enzyme to be any better. It was happy the way it was in its lab environment where we were kind of giving it a good chance at life. But it really kind of got me thinking about just how biased is our view of enzymes? Just how wrong are the textbooks? The textbooks are full of these examples of massive high activity, exquisite specificity, exquisite regulation, and we could never evolve that kind of stuff in our experiments. And the answer is that our view is incredibly biased. So Chelsea Vickers' lockdown project during the pandemic was to make this figure. This is a tree of life. Many of you are probably familiar with the idea. It summarizes all the genetic diversity of all the known organisms on the planet, mostly bacteria, not very many eukaryotes like us, the plants and the animals and the fungi and humans, and a few more of the archaea. And the rule of the game Chelsea played was to say, if a single enzyme had been characterized from a branch on this tree of life, she'd color that branch red. And the branches on this tree are hundreds of species. Each one of those species has thousands of enzymes. There might be 1 million enzymes in each branch of this tree. And Chelsea colored it red if one of the million had been characterized by a biochemist somewhere. You can see there's pretty vast swathes of black, where we just have absolutely no idea about anything to do with the biochemistry, the enzymes, the metabolism of a vast number of species on the planet. It may not surprise you to learn that we're incredibly biased towards knowing a lot about eukaryotes. Over half of what we know about enzymes is eukaryotes, and within that is a relatively uninteresting biochemically species called Homo sapiens that we know the most about. And so it struck me that we needed to do something about this. We needed to bring the knowledge of enzymes in line with our emerging knowledge of the diversity of all of biology. And Chelsea and Jordan and others have taken on this task. So for example, it's a pretty interesting black line here. And that's the one that includes archaea most closely related to us. So prokaryotes most closely related to the eukaryotes. Only recently discovered. Another way of saying that, I suppose, is that you could put this picture on your mantelpiece, because if you go back 100 million generations or so in your family tree, there's a pretty good bet that grandpa looks something like this. Discovered in 2020, and Chelsea and Jordan immediately jumped in there and decided to characterize some of the enzymes, because no one had ever done that before. And so, for example, in Jordan's recently completed PhD, she picked on one and did this beautiful kind of dive into the structure and function of this enzyme that had never been, from an organism where no enzymes had ever been studied before, and found that it was kind of weird. It does 3 jobs. The one in humans only does 1. But at the same time, all of that detailed biochemical characterization that she did let her it became obvious how this unusual enzyme that had never been glimpsed before could evolve into the type of enzyme that are in us and our relatives on the eukaryotic branch. So some real kind of fundamental look at this unglimpsed diversity at the molecular level. So I guess, in terms of this fundamental research into enzymology and metabolic biochemistry, the take-home message, in my mind at least, is that textbook biochemistry needs to meet the messy reality of biology. And in fact, rather than defining enzymes with characteristics like catalytic power, specificity, exquisite regulation, probably more accurate definition might be slow and sloppy, diverse, easy to evolve. I'll just have to write the textbook next, I suppose. I'll get on it. That's the fundamental stuff. For the last part of the talk, I want to switch and talk about how this knowledge of enzymology can be used in more applied ways. Here's a quiz question. So one thing about moving back to New Zealand and starting my own lab group is I realized how expensive everything is here in terms of lab reagents. And so molecular biologists and biochemists like me all around the world spend a fortune of Marsden money buying ultra-high value enzymes. So every molecular biology lab in the world will have a tube of enzyme, one of many enzymes like this one, that retail for prices like this, hundreds if not thousands of dollars. Here's one. I prepared earlier. And that's our discounted price. Whereas it struck me that I'd moved back home to New Zealand, to a country where what we did as the backbone of our economy was to sell biological products like this to the rest of the world. And so at the going price of $169 a tonne, you could buy almost 6 tonnes of logs, or you could buy this. One of these biological products, I posit, is a better economic backbone for our country than the other. An ultra-high value, near-weightless, exportable commodity. And so in parallel with all this fundamental stuff about enzyme evolution, enzyme diversity, we've leaned into this idea of trying to find more examples of ultra-high value enzymes that could be bona fide export earners for New Zealand, building the biotech community here. Obviously, not all enzymes cost that much. I bought this on sale for $11 from Countdown. Not on my P card. But there's a market that could be had. If you think I'm crazy, I just wanted a quick aside based on something I found while researching for this lecture. It's a company you might have heard of in Denmark called Novo Nordisk. So it was founded in 1923, 101 years ago, by 3 clever Danish biochemists, 2 of whom were a married couple, actually, as it turns out. I only learned that yesterday. So they figured out how to prepare and purify and formulate and sell a protein that you might have heard of called insulin. And fast forward 100 years of innovation, of hiring thousands of incredibly smart people, a constant focus on research and development. Last year, the headline was that the value of Novo is now greater than the rest of Denmark combined. And so my point, if you're in BTEC 301, I've stood on this soapbox before, we can do this. Denmark has roughly the same population as New Zealand. Denmark has a proud history of primary production. It's changed its economy to one that develops and innovates and exports things like this. So, what have I done about it? Not that, obviously. Otherwise I wouldn't be here. We have, however, tried to use enzymes for biotechnological purposes. And so back in the day, Robbie and Susan managed to, we licensed an enzyme to an overseas company called Enzymes, generated a little bit of royalty income at that time. Danny had this great PhD in collaboration with Lanzatech. So back when it was a little New Zealand startup company before it became what it is now, which is a multi-billion-dollar US biotech juggernaut. We've got a patent with them. For all we know, Danny's mutated alcohol dehydrogenase is in one of their production strains. Tiff, who I saw around somewhere earlier, is the world expert on this enzyme, RNA ligase, and we got really close to commercializing it. It would have been a version like this, an enzyme in the freezer of molecular biologists around the world, with an international company called PerkinElmer, who fired their business manager, got a new one, and was less interested in our product. But really, it's been the most recent work with Chris and Lee and others in this kind of cheap and cheerful kind of setting that has started my biotech empire, I hope. So Lee worked on a little test for winemakers. Winemakers need to do lots of tests in order to make good quality wine as cheaply as possible with the biggest possible profit margin, sell it more expensive than Australian wines. And so she made a test for the nitrogen-containing compounds in grape juice, something that winemakers need to know about. They need to know quickly. They want a simple answer. She designed this test using enzymes that were new to science, but built on technology that was started by Dave Ackley and Alistair Brown out there, VUW-owned IP, to give them what they wanted. Yellow, not enough nitrogen in your grapes this year. Blue, too much. So you can manage it accordingly. And assistant winemakers and winemakers around New Zealand, like this young guy in Blenheim, tested our prototypes. They really loved it. So I've got to the point where the reason I was in the lab on Sunday was not to take the photograph of the wine bottles. It was to work with Lee to prep our first sample of these enzymes for potential commercial sale. So we've just prepped, with Elisa's massive amount of help, this valuable cargo. We've prepped this magical blend of enzymes to effect this test, and we've just sent it, just arrived this morning, to a major wine supply company in Australia, where they're hoping to roll it out to Aussie wine growers over the summer season. And so it looks like a lot of white powder. Actually, less than half a gram of the white powder is the enzyme part. The rest is just filler, but we're not telling them that. And if this goes well and they sell the kits that they want to sell this summer, that'll be about $35,000 in sales for less than half a gram of enzyme that Elise and Lee made this month. Exciting times for us, and we're hoping to do a commercial licensing deal with this wine supply company. Not exciting enough for your average venture capitalist for whom wine is too small of a market, but it turns out that Lee was smart enough to realize that her enzymes could also detect related metabolites that are markers of chronic kidney disease. And getting into the biomedical kind of market for these kind of tests adds a lot of zeros on the end of your business modeling. And so what we're fleshing out now is tweaking the test in such a way that it might be in a form to enter a biomedical market. The back of the envelope calculations for this are kind of frightening in some ways. Make very conservative estimates about getting it into one of the big American pharmacy chains like CVS, Walgreens. They sell a few tests for a price, they sell similar tests. All of a sudden, that's a $150 to $200 million market in sales per year. And the fun part of the back of the envelope calculation is it would take about this much enzyme, about 2 kilos. And so we can make this in New Zealand. We could have an export market of those kind of quanta using the technology that we have here right now. I think that's something to hold on to. Of course, it's a fast-moving field. As I said right at the start, woke up this morning and Nobel Prize has been given in it. I just want to reassure you that we're on top of it. The two guys on the left, Demis Hassabis and John Jumper, what they came up with, AlphaFold, is already totally run-of-the-mill in our group. We've had students for years using that to predict protein structure production. The real genius on the slide in terms of protein structure, function, and design is David Baker. His amazing tools for designing entirely new enzymes from scratch that don't look anything like the ones found anywhere in biology are mind-blowing, and I'm very fortunate to have a new PhD student, Isaac Green, starting next month. And he's going to be using that to design our next generation of proteins to be involved in this RNA vaccine work. So it's exciting times, but if I was going to leave you with one image, I think it should be this one, that based on what I've told you, the future of New Zealand's export industry doesn't have to be dependent on things that we sell for $170 a tonne and put on ships to ship a really long way to get to the target markets. But instead, if you leave the bright future in the hands of Google's new generative AI app, you could make something on this kind of scale, but ship it to those offshore markets, something that looks like this, and that would be much more befitting. So finally, then, I really do just want to end by saying that, that the real great joy of being in academia is not giving lectures, it's not writing grants, it is hanging out with these guys. And so here's a selection of photos of group members through different kind of eras of it, I suppose. Everyone whose work I've talked about today is in there somewhere. And as I say, it's really all due to them that I'm standing up here, and I'm extremely grateful for that. Thank you.
Wayne. Super tempting to roast you at this point, but this is a very formal affair. So, tēnā koutou, tēnā koutou, tēnā tātou katoa. Nisa Bulavanaka, it's Fijian Language Week. I'm Professor Nikki Nelson, the Dean of Faculty of Science, and I'm delighted to give the vote of thanks. I feel very privileged to be up here to thank you for your inaugural lecture, Professor Patrick. Just making sure you're still on the screen in the video, so your dad can watch it later.
Right, great, thanks. Yeah, he'll be chuffed.
I acknowledge the Chancellor, I acknowledge the Provost and any other senior leadership in the room that I've missed. All our colleagues, friends, Monica. Thanks for coming. I'm hoping the students here haven't been bribed inappropriately to come with marks or anything like that. You can tell me later if that's the case. It's a real pleasure to have everyone here tonight. This evening we've been honoured to hear Professor Patrick share his enthusiasm for enzymes with us. His contagious excitement just reminds us that we're standing in the frontier of a really exhilarating field, as exemplified by the Nobel Prize today. What struck me most about Wayne's lecture is his vision for our future, the new technologies that are letting us study the structures and functions of enzymes from weird and wonderful microorganisms. Alongside that, we're witnessing this dawn of AI-driven protein design, and soon we'll have the capacity to create artificial enzymes tailored to catalyse whatever we please. And that is beginning. It's not a future that we haven't seen yet that's already in place. I think Wayne's showed us of his really ambitious efforts to bridge not just academic research, but to take it into practical application, and launching a biotech company to bring enzyme-based biosensors. That's just an amazing achievement, so well done. Thank you. Cutting edge work. But Wayne's forward-thinking approach is evident also in other areas of his work, in his contributions to leadership within the Faculty of Science, University, and the wider scientific community is enormous. I've really valued having his counsel as a Deputy Head of School and currently as the Associate Dean Research in our faculty. His dedication is to advancing his own work, but also those of his colleagues and his students, other researchers in our organisation is really valued, so thank you, Wayne. I note he's standing here quite nervously because I do actually have a reasonable amount of dirt on Wayne, but I promised that I would treat this occasion very formally. So the PC way of describing Wayne, and you can ask me later about what that really means, is an enthusiastic brainstormer of unconventional solutions, a critical thinker and a realist. But, you know, the Muppets and the guys that sit up on the balcony, Waldorf and Stadler. More over a wine later. So to conclude, look, I just want to congratulate Wayne on a fantastic inaugural lecture. Like I said, it's a real privilege to be up here doing this job. You should be immensely proud, not only of your presentation tonight, because it was super interesting and we were awake the whole time laughing, so... but just all the outstanding work of your career so far, and as a professor of this organisation, you should continue to contribute in that way, please. That's the end tonight. I welcome you all to come next door and have a nibble and a wine and chat to Wayne and just enjoy each other's company, and thank you all so much for coming. But at this point, let's just thank Wayne.